Okay. Hey everyone, this week we're going to start on our vegetative propagation experiment. And this is different from what we've done so far. So far we've talked all about growing trees from seed, which is known as sexual reproduction. You have a parent tree or a parent flower, male flower, and a parent female tree or a female flower. They, pollinate, do their thing, and you have a new seed and a new tree that is genetically diverse from both of its parents. With vegetative or asexual propagation, what we're doing is essentially creating a clone. So the new tree that we get is going to have the exact same DNA as its parent. So the way that we do this, we're doing what's called hardwood cuttings, which just means that we're taking the cutting in the wintertime. So it can be a softwood species such as this. Nope, this is not softwood. This is a hardwood cutting to get really technical. But in this language, what we mean by hardwood is that it's taken while the tree is hardened off, so in the winter. If we took cuttings in the summertime, when the wood is actively growing, that would be considered a softwood cutting. And different species prefer different things. So what we've got here is a bunch of different species, and this is a bit of a variation from how we normally do this experiment in this class. Usually we're pressed for space because each student is doing their cuttings. To this year, we have a little bit more space so we can all go along for this journey and try out a couple of different things. And it's exciting for us because we don't know what the results are going to be. So what we have here are some amber maple, we have high bush cranberry, basswood, balsam poplar, dogwood, pussy willow, apple, and black cherry. So we're going to prepare each of these sets the same way, and we're going to have a little bit of a difference in how we prep them. So we're running really two experiments here. One is, does this species do well with vegetative propagation? And the second is, does this species do better with what's called a rooting hormone? So we have a rooting hormone here. Usually it's a derivative of willow. So willow is just a tree species that propagates really well. And so some of these we expect that there won't be any difference between those treated with the hormone and those that are just, prepped and stuck in the substrate. But it'll be interesting to see for some of these species if there is a difference. So I'm going to show you how we're going to prepare them. Then we're going to prepare all these. We'll take them out into the misting bed, and we'll show you what the monitoring process is going to look like. But to prepare them, we're going to just help it along. Something like a willow, you can probably just stick this in water or in substrate or outside in soil, and it's going to root and grow. But to help it do so, we're just going to shave off a little bit of the bark here and expose that vascular cambium. Okay, remember that the vascular cambium is an area of active cell division, so it's meristematic tissue. And what we're doing here by exposing that cambium and putting it into a substrate is encouraging roots to grow. It can, malfunction and grow buds or leaves or something instead. We might have roots coming out of one of the active buds here, but we're just going to, give it everything we can and understand that these are biological beings and they don't always follow the rules. But by cutting the bottom flat, stripping some of the bark, that's giving it the best we can give it in order to encourage it to do what we want. And then we cut the top on an angle, or we have the top that has a terminal bud on it, and that's just to prevent the water from pooling on it. So that is really all that's required here, depending on the species. And then the ones that we're treating with the rooting hormone, we're just going to dunk into water, and then once it's wet, we're going to put it in our rooting hormone, and we're just going to roll it around. And we really can't get too much on there because once we take it out, we're just going to tap it, and then all the excess is off. So we can see that it's coated and it's ready to go. So we're gonna prep all of these cuttings will get the skin kind of sheared off the bottom. So we'll expose the vascular cambium on all of them. And then half of them we're going to treat with the rooting hormone, half we're going to just stick in the misting bed without. And once that's done, we'll get them in the bed and we'll show you what it looks like then.